Hello everyone, this is Deepti, Sanya Lecturer in Biotechnology. I am here to present a simple mass topic. It is nothing but the difference between bright field microscopy and dark field microscopy. Before that, I would like to give a little bit introduction part regarding microscope. Microscope is nothing but it is an instrument which uses lenses, a combination of lenses which is mainly used to magnify and to resolve the objects. And simply it is magnification is nothing but enlargement and resolution is nothing but to distinguish between two objects. So which in the simple words, why we are using microscope is nothing but the things which we can't see with our naked eye that can be easily absorbed under microscope. So for that purpose we are using this particular microscope. So if you observe this particular slide, so if the object size is 10 mm, then our naked eye can see with our naked eye. So if the size is so small, so that is 0.1 nanometer, then we can't see with our naked eye. For that purpose we require special type of lenses, for example normal glass lenses in the case of light microscopy and magnetic lenses in the case of electron microscopy are used. So as a result we can see with our such type of small tiny atoms, for example tiny atoms, amino acids and structure of DNA can be clearly visible. So what is the importance of microscope, why we are using nowadays? So our next one comes. There are two types of microscopes are available. One is light microscopy and electron microscopy. Light microscopy, here light is a source of energy is used, whereas in the case of electron microscopy, electrons as a source of energy is used. In the case of our present topic, dark field and bright field microscopy, they fall into light field microscopy category. So before that, I would like to give a little bit what are the parts which are present in the compound microscopy. So these are all the parts which are present in a simple light microscopy. So I am coming to this particular one. This is the eyepiece and this one is the body tube, and next one is nothing but course adjustment one, and this one is the objective lens, and this one is the stage where our specimen is placed, and this one is the normal condenser lens, and this one is the complete things which all the things can be maintained. So it is the simple parts of already mentioned light microscopy, all the things bright field microscopy, dark field microscopy, phase contrast microscopy and fluorescent microscopy falls into this particular light microscopy category. The description part of bright field microscopy. In this particular bright field microscopy, the name itself indicates that bright field means here the background is having the bright color, and whereas in the case of specimen that is darker in color, as and it is quite opposite in the case of dark field microscopy. In the case of dark field microscopy, the background is darker in color and the specimen is lighter in color. If you observe this particular type of slide, then you will know what is the difference between the dark field microscopy and bright field microscopy. Here, in this case of dark field microscopy, one special type of disc is arranged, so that is referred to as top, which does not allow the light to pass to the condenser. As a result, the background that will appear darker in color. But why we are getting the specimen or object in light color means only the diaphragm, it is allowing the peripheral light which is reflected by the specimen, so that will allow us to pass to the objective lens. As a result, we are observing lighter image. So here darker background and the image will be brighter in color, whereas in the case of here bright field microscopy, there is no such special disc is absorbed. So as a result, we are observing bright field and the object is nothing but it is absorbed in darker color. So this is the major difference between dark field microscopy and bright field microscopy. If you observe this particular slide, then you will know what is the difference between dark field microscopy and light field microscopy. Here, this is the normal representation of tissue paper when observed under both the things, and this one is the bright field one, and this one is the dark field one. So just compare this particular two images, then you will know the clear picture what I am trying to say this particular presentation. So here the background is brighter. This is in the case of bright field microscopy. Background is brighter, whereas in the object that is nothing but threads of tissue paper. So this is in darker image, whereas in the case of this particular thing, so this is example of dark field. If you observe this one, the field is darker, and the particular threads of this particular tissue paper, it is in the form of lightly illuminated one. So that is the major difference between dark field microscopy and bright field microscopy, what I am trying to say in this. And what is the significance why we are using dark field microscopy is nothing but dark field microscopy is a technique which is mainly used to observe the unstained samples. So as a result, the normal particular samples that cannot be contaminated with any type of additional chemical substance, which it is that can be possible in the case of bright field microscopy. If we are using bright field microscopy, here we need to add some special type of stains, nothing but chemical substance that will impart color to that particular sample. So as a result, if you are studying the particular composition of sample, we may not get the exact composition of that particular thing. So whereas in the case of dark field microscopy, here we are using unstained samples. So as a result, that can be easily recognized each and individual components of the particular things object. And if you observe this one, this is nothing but observation of diamonds, and in the case in the microscope, dark field microscopy, it just look like this. Here it is having the darker background, and the particular diamonds that will brightly lit against that particular dark background. So that is the things why we are using dark field microscopy. And sometimes bright field microscopy is also referred to as sometimes is also referred to as light microscopy. So it is that is mainly used in normal hematology department or any microbiology department in order to view the microorganisms like bacteria, but normal viruses. So that is the significance why we are using bright field microscopy. So this is the major difference. I hope all of you understood this particular major difference between what is the major difference between bright field microscopy and dark field microscopy. So thank you.